All right. Again, good morning, everyone. I'm Annalisa, the Education and Outreach Coordinator with the American Society of Gravitational and Space Research Student Chapter. Today, we welcome Alain Berinstain to this webinar. Alain is the Chief Strategy Officer at Space Tango, a company recognized as a leader in automated integrated systems for biomedical applications in low Earth orbit, headquartered in Lexington, Kentucky. Alain leads the identification of new markets in low Earth orbit and opportunities for automated in-space manufacturing for health and technology products, and corporate strategy, stakeholder relations, and space life sciences program implementation. He brings over 25 years of experience in both interdisciplinary science and technology and global business development strategy. He served as the Vice President, Global Development at Moon Express, and has also held multiple leadership roles in a variety of organizations, including the Canadian Space Agency as a Director of Planetary Exploration and Space Astronomy, Director, Science and Academic Development, Director of Policy for Canada's Minister of Transport. Alain holds a doctorate in physical organic chemistry from the University of Ottawa, a master's degree in space studies from the International Space University, and a bachelor's of science degree in chemistry and biochemistry from Concordia University. Again, let's welcome Alain speaking to us about unearthing answers, automated capabilities for biomedical applications in microgravity. Alain, take it away.
Thank you very much, Annalisa, and thank you for setting this up. Thank you to everybody for taking some time out of your Saturday to chat today with us about some of the exciting work we're doing at Space Tango, and anything in general that we'd like to bring up at the end. I'd be happy to field any questions. And thanks again, Annalisa, for making all this happen. Let me start by sharing my screen so I can use Annalisa, can you let me know if that looks the way it's supposed to look?
That looks good.
All right. Thank you again, everybody. And I'm going to go through these slides, and then we will have some questions at the end. So I think it's easiest if we save your questions or write them in the chat window, and we can deal with them when we get to the end. So yeah, I'm Alain Berinstain, and Annalise asked me to discuss a little bit about myself, although she just did most of a good introduction about my background. But born and raised in Montreal, Canada. I have a bachelor's degree in chemistry and biochemistry and a Ph.D. in physical organic chemistry. I also have a Master of Space Studies from the International Space University, and I'd be happy to answer any questions about that. But it was key in helping me pivot towards a career in the space sector. I spent most of my career so far at the Canadian Space Agency, and Annalisa listed some of the positions that I had. And mostly it's about science, space science program management. I did take a little detour for a little bit. There's an error. There's 2015 to 2017. I was with the Minister of Transport, his Director of Policy. It looks a little weird, but there is a link. The Minister of Transport in Canada is, or was at that time, Marc Garneau, who happens to be Canada's first astronaut. So it's not a coincidence, or it's not weird that I was there. We've known each other for a long time, and when he got that position, he asked me to come and work with him and do some non-space work for a couple of years, which also is very relevant to all kinds of work that we do today. I was with Moon Express for a couple of years, and now I am with Space Tango, and just like Annalisa said, I do thinking about our future and setting us up for the future. I take our CEOs vision and turn it into a strategy of how we're going to get from where we are to where we want to go in the long term. And today I'm going to tell you about our systems, our capabilities, and who we are. But typically, in a nutshell, what we do is we're well known for our automated solutions in low Earth orbit, and we have settled in on a lot of health and technology research manufacturing using microgravity mostly on ISS. We design hardware, we certify hardware, we operate hardware on ISS, and we're in the family of implementation partners with the ISS National Lab. And what we do a lot of is working with researchers who want to do research in space, watch them do what they do on the ground, and then turn that into something that can be done in microgravity with our analysis and our hardware solutions. We do rely on a lot of automation. It is something that is at the core of everything that we do. There are pros and cons to this, and again, I'd be happy to get into that. But it has really worked out well for us, and we can automate and remotely control our experiments from the ground. So this is the only slide here. I think this is a space-savvy group, but why microgravity in the first place is on the ground, there are some physical characteristics that occur that are masked by the forces due to gravity. For example, some gravity-driven convection can happen on Earth when there are different densities. That does not happen when we are in microgravity. Sedimentation occurs on the ground that does not occur when we're in microgravity. And buoyancy is the same. So there are some limitations to some systems on the ground, and they go away when we go to microgravity. So how do we use that? How do we use that for research? How do we use that for manufacturing? If you think about some manufacturing processes in any field at all, in any kind of sector, in a lot of manufacturing processes, you have to wait for stuff to settle or stuff to float, or you have to mix it, or you have to make sure that it stays mixed so that it doesn't settle. All those kinds of processes are dependent on gravity one way or the other. Sometimes gravity helps, sometimes gravity gets in the way. There are some biological processes that are changed when we go to microgravity. The gene expression changes, and distribution of materials changes. And so all these interesting factors that occur when we go to microgravity is what we like to explore and also leverage to get to where we want to go and do what we want to do. At Space Tango, this might be out of date by one mission or so, but we've flown to ISS at least 25 times. And each time it's with multiple payloads, multiple experiments. And so we can really proudly say that over the 6 or 7 last years, we've flown around 200 experiments on ISS. So we're pretty experienced when it comes to the family of implementation partners that way. We are based, like Annalisa said, in Lexington, Kentucky. We do have an office in Florida, actually, and that's where I am based, in our office in Florida. And we have an office in Houston as well. Most of our launches are out of Cape Canaveral, and so we do spend a lot of time as a team. Even the Kentucky team comes to Florida quite a bit. Every time we launch, we do those final integration processes out of the Florida facilities and out of the NASA facilities in Florida. Part of our system that I'm going to tell you about is based on our architecture, which is based on a CubeLab architecture that gets sent to ISS. These are some cartoons of our CubeLabs. It's hard to tell the actual sizes of these things, but they are more or less equivalent to, if you compare to CubeSats and the nomenclature there, a 1U CubeSat or a 1U CubeLab is about 10 centimeters by 10 centimeters by 10 centimeters. Double those up, you get 2. Put 2 of those 2s together, you get 4. Add another couple, you get 6, and then you can go up to 9U. We do most of our experiments in 6U and 9U formats. but it is part of our experiment standard, is to basically stuff everything you need to do into one of these cube labs, and then all we connect to it is power and communications, and then we can control everything that's going on inside the cube. There are great advantages to this. These are certified by NASA to pass the safety systems in terms of containment of fluids, for example. And so we can change quickly what happens inside from one flight to the next, and it still meets NASA requirements. So that's one of the practicalities of using this design, this architecture. These are some real images from ISS. So on the right here, you see a 9U cube lab in front of a typically sized mid-deck locker, but this is our version called the Tango lab. So in our architecture, we have 2 Tango labs that live on ISS permanently, and the cube labs are hosted by the Tango labs. And you can fit 2 9U cube labs, kind of lengthwise, into each of these Tango labs. And again, in our automated systems, we don't use crew very much, but where we do use crew is to move these cube labs basically from the transport vehicle into ISS and basically plug them into the Tango lab for power and communications. And then they operate, and then when they're done, they get taken out by crew and sent back home. Now, I said we do a lot of work in biological systems, and what that means is often that these biological systems need to be continuously maintained in an environment that maintains the cell health or the biology health. It typically means it has to be incubated at a certain temperature, and sometimes even media has to be changed, which would mean that these cube labs kind of have to be kept powered, even from the ground when we load them, through launch, through transit, and through docking. And we do that in what we call a payload ascent utility locker, or PAL. And a PAL is just another version of a Tango lab, but is meant to be the container in which these powered cube labs are transported and moved from Earth all the way to ISS. And then actually the whole PAL is plugged into an express rack. Instead of the cube lab being installed in a Tango lab, it stays in the PAL. And there, like I said, the advantage is that these are designed to be continuously powered all the way up and all the way down, because in some cases, biology needs to be tended to. And it's not always biology, it could be other things. But for any application where you need continuous power, because if you waited the 3 days or 2 days between loading and actually plugging into the Tango lab, your biology could be not viable anymore by the time you get there. So the PAL is a solution for that. And we're always evolving these systems and making them better or with more and more capabilities. Another aspect of our capabilities, and it's linked to the automation, is that we can control all these things from the ground. I'm going to show you some of the cool things we can do from the ground. This is done through our Mission Operations Center. This is a picture of our Mission Operations Center and our Director of Engineering, who is sitting at the ops station in our ops center in Lexington, Kentucky. So we can control everything live. We receive telemetry. We can even talk to the crew directly through the systems that we are approved to do, certified to do, through our ops center. So we can monitor 24/7, we can intervene, and we can coordinate with any ground studies that are going on at the same time. So a lot of the work that we do is working with scientists or researchers on the ground and translating what they do on the ground to what can be done in space. Questions like, how do I contain my sample? This picture here is a picture of us loading some fluids into our equivalent of a 12-well incubation system. On the ground, you put a few milliliters of media or in cells at the bottom of the well, and sometimes you cover it and then you let it do its thing. You don't worry about that material floating out of that well? Well, we have to worry about those things in microgravity, and that's the translation that we do. Show me what you do on the ground, and then we'll translate that to something that's feasible in space. The conditions on launch are important. We have to ask, what configuration can it be in? Does it have to be powered? Does it have to be temperature controlled? And then in order to keep your cells alive, if that's what we're doing, what's the sequence of events that has to happen, or the timing in order to see what you need to see? What temperature do you need to keep things at? What's the gas mixture it needs to be in, the pressure? And what imaging needs to be done if there's any imaging to be done. Sometimes you have to sample what's going on, or you have to fix the cells at a certain time. All these questions are what we need to go through with our scientists. Excuse me. Excuse me. Another aspect of what we do is that we do everything sterilely. This is a picture of Trevor, one of our engineers who has been trained in sterile technique to be able to work in biological systems, in our systems, to be loaded and then flown where there can be no bacteria, no contaminants at all in our systems. Otherwise, you could lose the science. And so we've gotten pretty good at that. We can fly, here we're talking about biological systems in little cryovials, in tissue chips, in weld plates, and cell culture bags, and I'll show you some examples of each. This is our cryovial system, where these are little vials, little 1 milliliter type of vials, and we can launch like 600 of them at a time. This is for passive experiments, where the requirements for whatever has to go on inside this vial is taken care of with, all we need to do is keep them at a constant temperature. We don't have to change any fluids. We don't have to do anything to the sealed vials. We just need to get them loaded, get them up in space as quick as possible, leave them there for whatever period of time we need, bring them back down. And that's a great system for high throughput for simple systems that way. We've flown a number of experiments using stem cells, even brain organoids that we've flown with a number of partners, including UC San Diego and a number of others, including Cedars-Sinai. This is our tissue chip system at the top left that hosts tissue chips. There's a lot of science that gets done these days in these microphysiological systems, and they have very small amounts of fluid that flow through these tissue chips that will expose them to different experimental conditions. We can host most kinds of custom tissue chips, and most tissue chips are custom. And we will supply the fluidics that go in and out of each of these chips, keep them at the right temperature, and image them if that's part of the requirements, and fix them if required. Our systems are sterile, biocompatible, and we design custom fluid paths for each of the systems. Again, we've flown this a number of times with a number of partners, a lot through the NIH Tissue Chips in Space program, and through the NSF Tissue Engineering program as well, with a lot of partners from around the US. This science interface system is our well plate system. So if you've done any lab work, a 12 well plate system is a standard in labs, where you can fit 1 or 2 or 3 milliliters of fluid in each one of these little round wells. And the magic happens in the wells, and you do some media exchanges sometimes to keep the cells alive. You can do some sampling, some fixing. We have a version of it that will meet the constraints of doing these things in space, like they're individually capped, they don't have any air bubbles in them, and we can fluidically address each one of the 12 wells independently. And so you can expose them to different conditions, and of course, all done sterilely with biocompatible materials. And we've flown them on a couple of flights now, and it's a great way to do some experiments. We've again flown with UC San Diego on this, and some NSF tissue engineering projects as well. In this science interface system, we grow cells in bags. It's yet another way that some researchers do their culture. In a bag, with or without a support in the bag. You can have a hydrogel support, you can have a sponge support. And it's not too different from the others. Again, we'll keep them on a plate, and that'll be kept at the right temperature. And then they will go through their protocols. There's an inlet and an outlet for each of the bags, so we can do media exchanges, etc. A little example of some of the complexity of what happens in these boxes is in this top left. It shows you where the person's finger here is where the two bags would sit while they are incubating, and we can image those, and I'll show you some of the pictures. Some of the other stuff that happens in these cube labs, though, is important. Over here on the top left, you'll see what looks like a Yeti flask. It's an insulated flask that contains cold fluids, because media to feed these cells often needs to be kept cold until use. So within the same cube, we'll keep your cells at 37 degrees and keep your media at 4 degrees. And then when it's time to get media out of here, we will pump it out, warm it up before it goes into your cells. As fluid comes into your cells, there's some waste that has to go out. And these are bags that collect waste. And that waste sometimes is truly waste, and other times it still contains cells that have some information that scientists want to have. So you can have some fixatives that are preloaded into these bags, and you can have a number of different waste bags depending on the sources of that waste. And again, we can control all these fluidics remotely and in an automated fashion. We've flown this a number of times with a number of partners, including our friends at UC San Diego Sanford Consortium and Cedars-Sinai as well. One of the great things we can do is image what's going on inside these cube labs. These are some examples of some images taken just with a regular high-resolution 1x camera. With different optics on it, you could get different types of images. On the left, you'll see an image of a tissue chip from our partners at Emulate, and this was imaged using a fisheye lens. This is one of those wells from the 12-well plate with some organoids in it from UC San Diego. And it's just because it's a high-resolution camera, we can zoom in and get some of these images, and just the shape of some of these cells or collection of cells. These are organoids in this case, gives information. And here's another set of 1x images from the Cedars-Sinai payload. And here there's some information about how the cells gather together, how different groups of cells clump or not, and general health of cells to the trained eye can be elucidated just from these images that we bring down live. We can tell what's going on, and because it's live, we can adapt, change the experimental conditions, do media exchanges, as if it's almost like you were right there. One of the most powerful analytical techniques we have is through microscopy. We have this really fun system that allows movement in the XY stages to move the objective of the microscope over whatever part of the experimental plane you want to look at, whether it's over a different well or a different part of the bag. Then we can do phase contrast, bright field, or fluorescence imaging, and even capture video through the microscope. And this also has a capability to do Z-stacks. So we can do full X, Y, and Z planes for imaging. And to give you some idea of what happens, on the left, you'll see, and I don't know how well this is coming through to the viewers here, but On the left, you'll see some twitching muscle bundle fibers from the University of Florida, looked at through the bright field microscope. These are muscle bundle fibers in tissue chips that are being electrically stimulated, and they're contracting due to the electrical stimulation. And the scientist, Dr. Mullaney, gets a lot of information about this. She compares ground to Earth samples, and one of the phenomena that happens to some biological systems when they go in space is that they demonstrate accelerated aging. So she's looking at accelerated age. She's looking at aging in muscles from humans in these systems. Fascinating stuff. On the right, you'll see some fluorescence imagery from our fluorescence microscope. These are images from our friends at the Sanford Consortium at UCSD. We can do actually 3 different channels of fluorescence, but this is the red and the green. We can put them together and create these wonderful images of cells, and this is the bright field where you can actually see the sponge support where it is. On the right is an animation of Z-stack, slicing through the 3D structure of the cells during the experiment. And again, the researcher, the trained eye, can see what's happening here in terms of the evolution of the cells from one phase to another, monitor these things, and even react if necessary along the way, and compare what's happening in space to what happens on the ground. So overall, we can provide a lot of the same types of environments that researchers need to carry out on the ground, but we can provide that in our cube lab in our own way, kind of thing. And that's where our expertise comes in, is the ability to do that. We can incubate at a target temperature. We can store media samples at a lower temperature, all within the same cube. We can maintain a gas environment, because a lot of these systems require a specific gas mixture. We can do media exchanges, we can do media sampling, we can do endpoint cell fixation, imaging and microscopy I talked about, electrical stimulation. And we monitor all this real time through our Ops Center and a customer dashboard, which is really cool. These are just some screenshots of what our partners can, basically, they can view what we view live at the same time as we do on their phones as things are happening. They can see the temperatures that are happening. They can see some of the imagery. And there's a lot of data there to be looked at on a continual basis. That's what we do today. Where are we going in the future? A lot of the systems I talked to you about were really our operational business that we run today commercially for our partners out there. But what this has done is created a great set of partnerships where we can create, it stimulates some wonderful conversations with these partners about where is this all going in the longer term. There are some, I described to you how we do a lot of R&D right now in space, and it works, and it works well. It's a lot of work. It's a lot of work to translate what we do on the benchtop to what we do in space in these little cubes. Our vision for the future is to do some work in a more glove box, more benchtop type of environment. And we call this the Tango box, which is in development right now, which will be a glove box environment, typical, that kind of exists today on ISS, that can be used in that mode, but it'll also be much more advanced than that. And that it'll be equipped with a lot of robotics, automation, and onboard analytics. You know, a lot of times we need to return the samples from ISS because we need to analyze those cells. It's hard to do that within a small cube. Imagine that a lot of those analytics are done on board, and that the cells don't even need to be returned, which increases the efficiency of access to space by quite a bit. There are also some modes of using microgravity, like I was talking about at the beginning, to make things in space that you can't make on the ground. And we're very, very focused on this in the long term, in identifying some of these applications for the use of space for manufacturing in space. We do have a great project ongoing with our partners at Lam Division on artificial retinas. We're looking at some organ growth, the regenerative medicine using stem cells and organoids. We're looking at making some nanomaterials in space, or using nanomaterials in space to make other things. And to provide services also to the commercial space stations that are coming up. They're going to need machines like this to be able to provide services to their own customers. So we're very, very involved from a strategic perspective in what happens next. Our business model involves continuing to do what we do today, but also transitioning that to the commercial space stations that are coming online, and also doing more and more use of microgravity to make things in space that you can't make on the ground, or that you can make better in space than on the ground. We've been working also with some of the commercial space station developers. We have some MOUs in place, and we have some really interesting discussions about what we could do in the future. And this is, I hope this comes through okay, this is an animation our friends at Sierra Space put together, looking at their life habitat in the future and how it could house some of our Tango boxes. This is a mode where you have the crew involved in a traditional sense, and then we have some others that are doing things in a robotic way. Now, in real life, they won't flail around like this, but this is just to show you an idea of the functionality that'll be in there. Or we have Tango Labs on the bottom, a Tango box on top, and they can work remotely and automated. And in this manufacturing mode with these last two, they're actually working ganged up together, where something great happens in one, pass off the samples to the next one, and it continues the manufacturing process there, or the analytical process there. So it's just an idea of how things might come together in the future. I mentioned some work we're doing with Lam Division in making these artificial retinas. These are materials that will be implanted in people's eyes to treat people who have lost their vision due to degenerative diseases like retinitis pigmentosa and age-related macular degeneration. These are thin film materials that are made up of a photoactive protein that translates the incoming light into a potential difference that pumps protons from one side of the retina to the other, excuse me, and allows people to see what they couldn't see before. We need microgravity to do this, to increase the quality of these artificial retinas. Excuse me, and to be able to implant this. So this is a long-term project we have in work with them. We've already flown a number of times in our cube labs, and eventually we'll be using our Tango boxes to do this. We also have some really interesting 3D technology, 3D printing technology in development with some partners at Lawrence Livermore National Lab. This is where we're using we're developing a volumetric additive manufacturing technique for use in microgravity. They've developed the basic technology for use on the ground, but this is a really interesting technique for using in space because it'll actually work better in space than on the ground. You can have 0D printing where it's point by point, where you use a laser to build up a 3-dimensional object by building it up point by point. You can also use a 3D printing technique where you print filaments, which is a kind of a 1D technique where you build up a 3D structure by extruding filaments over top each other. You can have a kind of a continuous lithography process as well, or layer by layer, or continuous in a 2D at a time by shining light in a 2D array and then building up a 3D structure that way. But in this technique, we're talking about printing an entire 3D unit in one shot. There's a Pikachu on the bottom right that's been printed, and to give you an idea of what happens in this technique is that you completely get rid of the step artifacts that happen in more traditional 3D printing. and you can print the entire element at once. So this is just a quick animation of what happens. Is you take the object that you want to print, you create projections of it, 2D projections of the 3D element as you rotate it, either in a computer or in real life, and generate these 2D images from different rotations. You then basically put them in a projector and project them back in a movie into a vat of resin that is rotating at the right speed. It's all synchronized, of course, to recreate that 3D object as that light is being shone onto the vat of resin. You then drain the resin, rinse it off, and you're left with the 3D object that is not built in a stepwise step at all. It's the whole object at once. That was an animation. This is the one actual object printed that way. And if you look on the top right here, this is real life printing of that same object, not an animation. And the other great thing about this is that it goes super fast. This is seconds at the top left. So this happens in a minute. Again, unheard of in typical techniques. You can do other cool stuff like overprint. That screwdriver you see, as you can see, you can start with a shaft within the volume and then print the handle around the shaft. You can do all kinds of really nice stuff. And again, microgravity helps. And I could do a whole hour just on this, but I won't. Just to wrap things up, that kind of brings it all together for us. We have three main business lines that we're working on. Contract research is what I spent most of our time talking about. And it's a great use case pipeline for us to figuring out what we could manufacture in space. We're going to be providing services to space stations and the expertise that come with it, and providing tools to them that will help them meet the needs of their customers. But where we really see the long-term value in our company is combining these two capabilities to manufacture in space for addressing large terrestrial markets. So that's me, that's that, and I'd be happy to entertain any questions that anybody has.
Thank you, Alain. Opening the floor to questions, but I'll start us off. As a researcher and as a potential partner, how can you get involved and have Space Tango as an implementation partner?
It's quite easy. Send me an email and we can go from there. But go to our website. You can see some of the work that we do. There are some contact points of contact listed there as well, and have a conversation, and we can do that. A lot of solicitations are out there on a regular basis from NSF, NIH, the National Lab, that all encourage researchers to reach out to implementation partners, and we'd be happy to talk to any of you.
To fill in the silence here a little bit, oh, we have one question come in on the chat. Any comments on how the disturbances in microgravity on the ISS affect the experiments and how it is addressed? Sure.
So when we talk about disturbances on ISS, there could be a number of different ones. So let's talk about environmental first. It is a shirt sleeve environment on ISS, and so it's expected to be ambient temperature, but that can even change with time. So how do we address that? Is we provide inside the cube a very carefully temperature controlled environment. So that's one example. Now, one of the reasons we go to ISS or to low Earth orbit is to be in a microgravity environment. Microgravity environment is kind of easy to talk about, but it's actually quite complex. What do we mean by microgravity? Why don't we say 0G? We say microgravity because it's actually not zero. Why is it not zero? There are a number of reasons about that, but some of it is vibrations. So anytime there are vibrations on board, even from a nearby fan, that disrupts the gravity levels that your experiment feels. If you shake or if you stir, that disrupts the level of gravity that your experiment feels. So there are a number of ways to address this. There are in still on ISS, I believe, but there have been in the past microgravity isolation systems. This is where, for example, your experiment platform can be physically isolated from the structure of ISS. And it does it through padding or even sometimes magnetic levitation. So that if an astronaut goes by and kicks your rack just to be able to move through the station, it won't disrupt your experiment. So there are a number of ways to address this. So it depends what it is, what disturbance we're talking about, but disturbance in the gravity levels itself has to do with the careful experimental design, making sure that there are no fans nearby. But also, you need to understand, it's more complicated than that. You need to understand what levels and frequencies of induced gravity around you your experiment is susceptible to. And that's what you need to address. If you try to address all levels of microgravity, all types of things that happen, that's a hard thing to do. Even the ISS itself needs to be boosted once in a while to correct its orbit, to make up for the little bit of drag that is up there. When that happens and one of the booster engines comes on, there's added gravity to everything that's attached to the space station. And that can be felt by some experiments. So those are all really important things to keep in mind as we're designing an experiment and designing the environment that it's in.
Everyone is also welcome to give us a message in a chat and I can read it. From Chirayu, does Space Tango plan to employ systems that can recapitulate artificial gravity to mimic gravitational levels on Moon or Mars, somewhat similar to JAXA and what they are doing on Kibo module? As we expect, we will require a countermeasure to protect astronauts for deep space travel.
It's a good question. So just in general, just to kind of repeat where this is coming from is we go to microgravity, but that gives us the microgravity environment, the low Earth orbit environment, but the world is planning to explore the Moon and Mars at 1/6 or 1/3 level gravity. Can we use ISS to create reduced gravity environments? And yes, you can do that using centrifuges that spin at the right speed, and the centrifuges are at the right size to reproduce that level of gravity. And in fact, sometimes you want to reproduce 1G in microgravity. Sounds a little crazy, but when you really, really want to do a very careful experiment where you want to compare 0G to 1G, you want to do it in the same place at the same time. And so having a 1G centrifuge right there with the exact same types of samples, the exact same hardware, the same place and same temperature and same everything, if the only difference you want is 0G to 1G, then that's a great way to do that kind of control experiment. So the question was about Space Tango specifically. At this time, we do not have that centrifuge capability or that artificial gravity capability, but it is something that is in our long-term plan. So you can imagine our Tango box could have a centrifuge. Sometimes you need a centrifuge just for cellular systems to be able to separate cells. We might need to do that. But you can also create the 1G simulated environment for the control, reduce gravity, and we'll be able to do that in our future systems. Any other questions? Well, Space Tango is a great place to work. We do hire interns. It is a great way for us to recruit as well. You can check us out on at our website, and you can look for opportunities there if you're still studying. If not, we're also always looking for qualified people. And we'd be happy to talk to any of you at any time. I'm lucky also to work with Annalisa, who is wearing her ASGSR hat today, but she does work with me at Space Tango, and it's a wonderful thing. And the fact that she's active in ASGSR is a great link between us and the general community. We do go to events like the ASGSR conference and others that happen as well.
All right, if there are any other questions, let's end the webinar here. Again, thank you, Alain, for taking the time to speak to us about Space Tango capabilities, and we'll see you guys at the next webinar.
Thank you, Annalisa. Thank you, everybody.